Welcome to this great opportunity we have to have Benjamin Voight, PhD, speak with us regarding human genetics approaches to identify variants and genes contributing to diabetes. Just a short bio, and thanks again, Ben, for joining us. Short bio for him. He was advised by Dr. Jonathan Pritchard, now at Stanford, and Nancy Cox, now at Vanderbilt. And in 2006, he received his PhD in human genetics at University of Chicago. As a postdoc research fellow from 2006 to 2009, he worked with David Altshuler, now at Vertex, and Mark Daly at the Center for Human Genetics Research at Mass General. From 2009 until 2011, he remained at the Broad at Harvard and MIT as a research scientist. And then in September 2011, he joined the faculty ranks at the Departments of Systems Pharmacology and Translational Therapeutics and Genetics at UPenn, Perelman School of Medicine as an assistant professor, becoming associate professor with tenure in 2017. He's a member of the Institute for Translational Medicine and Therapeutics and for the Institute for Biomedical Informatics. Sorry, I'm just trying to let people in. There's a lot of people joining us. And we're really excited to have him walk us through his approach and his newest work. So thanks again for joining us.
Thanks, Monica, for that introduction. Can you hear me okay?
Yes, perfect.
Great. Okay. No, great chance. Thanks for the opportunity to share my little slice of life and the problems and challenges that I think about. So I'll just jump right in. So I don't think I have to belabor to this audience that type 2 is the focus of my work, and this is a disease that is increasing in prevalence, and lots of reasons for that. Diabetes is complex. Genetics, environment, it certainly play a role. It's certain that the obesity epidemic has certainly contributed to that. But it's complex. It's not just one factor that contributes to risk. And I think a lot of the community is interested in trying to fundamentally understand the causes and consequences. In particular, I think from my point of view, and I've grown to appreciate having moved through the field a little bit, where maybe 10 or 15 years ago, we were really excited to find any genetic risk factor that underlied disease. As we'll talk about today, we are well beyond that, and we're trying to move into understanding really fundamentally what that means for the consequences of having diabetes, because heart disease, neuropathy, kidney disease, those are the really fundamentally debilitating comorbidities that really reduce the quality of life for individuals that have it over their life. And so if we can try to understand what the causes of those comorbidities are, I think we can start to really thinking about making progress on prevention. And so what, I start really from this point of view as sort of like basic scientist thinking about genetics. And what ultimately, I love the idea of trying to like tell stories about the genome, where we start at some level, really at the fundamental, like molecular biological level, thinking about gene regulation and the processes which are cell type specific. So trying to tell a story where like, oh, we discover a region of the genome that matters. That region of the genome influences regulatory factors that promote or repress transcription normally, but in a disease state do that in a way that's not that's disease predisposing. And if we can understand the choreography of regulation at that site and multiple sites in the genome, we can start to unpack the complex regulatory biology that underlies disease. And maybe, just maybe, if we can connect that to tissue-specific effects, we can understand more about sort of like the fundamentals of disease, ultimately understanding causal biology, but ultimately thinking about how we can perturb the system in beneficial ways. And not surprising, so if that's a fundamental question, that causality that leads us to therapeutics, how are we going to get there? And again, you have me here, and many of us have bias. All scientists have biases. My bias is, of course, human genetics. I think that human genetics is sort of fundamental. It gives, any particular observation we find in the genome tells us a little bit about that story. It's a root cause of disease. It gives us the opportunity to make causal connections between exposure and outcome. And I think that when we build those links, I didn't come up with this, but many people have sort of argued positively for the idea that if we have human genetically motivated and validated hypotheses for therapeutics, the chances of success for developing those therapies just goes up dramatically. So my bias is this. It's not the only bias you could have in this space, but that's where my thinking comes from. So all the work that I'm going to tell you about today really is fundamentally thinking about human genetic variation and how we can use different collections to try to walk through what I'll call this like roadmap to discovery. I think ultimately, if our goal is therapeutics, there's sort of like a pathway that we have to use to get there. And again, I'm viewing this from the human genetics point of view. You could have entered this sort of route in a different place depending on your point of view. But sort of I start with the idea of like, let's identify all the places in the genome that matter for diabetes. Once we have those, we should probably figure out what variants cause diabetes out of all of them that are possibly linked with the disease. We should connect those causal variants to particular mechanisms and the tissues of action. So do the variants change expression? Do they alter protein expression? Are they coding variants that disrupt the function of a gene? And which tissue or tissues do they operate in? That's fundamental. Once we are armed with that information, it's not enough. We have to translate that into model systems work, either at cell culture model systems with pluripotent stem cells, cell culture models, even model systems, zebrafish and mouse that are at high throughput, CRISPR screenings to validate non-coding variation. There's a whole catalog of things that follows from that. But my claim is that I think those experiments are much more operational, armed with human genetics information. And then once we have that, I think we can start bridging that to connect to complications of disease. And then ultimately that drives the directionality and gives us targets for therapeutic development. So to me, this is kind of like a roadmap. Maybe it's not a particular path that has a single arrow that focuses everything. You could bind in different places. So what I want to talk to you today is just sort of like 2 or 3 of the points that we're thinking about along a couple of these axes. So one of which is a large collaboration of work, and I'm grateful and excited to have interact with a great number of colleagues around the world, the UK, Germany, Mass General here at Penn, where really the goal is to try to like take our understanding of the discovery type 2 diabetes to the next level, armed with what I affectionately refer to as like the biobank era. And so we live in this like unprecedented time now where when I was a lad growing up studying diabetes genetics, really it was like we were struggling to get samples. It was like on the order of like thousands of cases, thousands of controls. We would struggle to do our scans for finding genes for diabetes, and we would fail, but then we would find like our friends and colleagues at other places. They would give their meager samples, we'd combine them together, and that led to initial suite of discoveries and sort of set the field going. We weren't the only one to come up with this. Many people studying cardiovascular disease, psychiatric disease, anthropometric traits. This was kind of the game plan. But in the last couple of years, that's changed dramatically with sort of the connectivity of electronic health records to mass scale genotyping and sequencing. And I've listed here just like a collection of examples of those biobanks. It's definitely not all of them. All of these contributed to sort of our effort to kind of assemble large-scale type 2 diabetes association analyses. But they all kind of play this nice game where they've hundreds of thousands of subjects have been genotyped or sequenced. We can phenotype them for diabetes or other traits using extensive health records data. The data and phenotyping is a little messy, so you pay a little bit of a price, but the advantage is that there's just tons of biobank data that can be integrated and synthesized. And so we did that here in this effort, and that ultimately culminated in, I think, like almost 400,000, a little over 400,000 patients with diabetes in a total sample size of about like 2.5 million with like 2 million controls. It involved a large number of efforts, so not just one cohort, but lots of cohorts, over 130 studies in total. And I think the other valuable point here is that, sort of like a painful point on human genetics generally is the focus on sort of monophyla of ancestry. And it's gratifying to see another advantage of these biobanks is that they don't just sample, they take advantage of everybody that accesses healthcare, and healthcare should be accessible to everyone. And so that allows us to increase the diversity of our representation from these studies. And so, over 40% of the individuals here actually represent non-European ancestry, which is great. And so when all that comes together and we pull the data together, do meta-analysis, we expand the number of loci that we discover. 145 new loci, 1,300 signals in the genome. So really emphasizing the idea that type 2 is a polygenic trait with many sites of the genome that matter, that we can discover these by expanding sample sizes and expanding who we combine together. And so, okay, that's great. And then you might say, oh, that's a little boring. I mean, you're just doing GWAS. So like, what can we do with this information once we've assembled it?
And can we back up just for one
Yeah,
second?
sure.
When you're looking at all these different repositories, can you, I don't know if you can comment on this, but can you comment on what, maybe which ones carry significant levels, sorry, I just had to let someone in, significant levels of T1D data? And also, how accessible are these biobanks to the typical, scientist? Or do you have to have sort of, a special admittance?
Yeah, that's a great question. For the first one about type 1, I think that definitely is biobank dependent. So for example, we were just talking about this in the call. I'm most closely connected of all these to the Million Veterans Program data.
Mhm.
And so that cohort definitely has type 1 diabetics in it, but you might imagine because the population of individuals are veterans, that's probably slightly lower than what you might have thought the prevalence of that disease was in generally if you were doing unbiased collection and prevalence assessment. But there's definitely data available in that, and I suspect different cohorts have different access to that. So like maybe like UK Biobank definitely has that, and those data have definitely been integrated into latest meta-analyses. I think many of these other cohorts have, but not all of them. So longer term on that trait, I think there's probably a good place to identify cohorts that haven't contributed, collect those phenotypes, and kind of build even larger meta for type 1. Now, on
Yeah.
the question of like access, yeah, I think different cohorts are differently good at this. So like UK Biobank is probably like the pioneer of like open accessibility with people who write project proposals and get access. And so they've been pioneers in that. Different biobanks like MVP, right, those data weren't consented for full public release of individual level data, so they literally can't be shared individually. But those, being involved in that project, there's an active effort for all analyses that are generated to create summary statistic repositories that people can access directly. And I know that there's a larger effort for the NIH to allow for investigators to do analyses with MVP data too. So there are vehicles, for example, to try to build those directions, but that's like very, very, very new. It hasn't always been that way, and I think different cohorts have different rules of engagement for those things.
Yeah.
If that makes sense.
So for instance, like FinGen, obviously Finland has the highest incidence of type 1, or one of the highest incidence of type 1. So that would be a very nice place to go, get into the genetic sandbox there. But how accessible is FinGen to just this sort of, early career scientist or trainee?
Yeah, that's a great question. And I know so like many of the things, it's sort of like if they, FinGen, I think, has tried to make some collections of summary data available. So like you could phi code T1D as a phenotype and analyze it. And I can't remember if FinGen has been trying to do that with a range of traits. I don't remember if I haven't looked at type 1, but I know they have a repository of summary data available. So that might be a place to look. But even if it wasn't, you know, different people are also differently collaborative. And so my suspicion with a place like FinGen is if you reached out to the people and said, hey, is there T1D data? Can we collaborate to try to build that together and try to learn something? It might be hard. The road might be a little bit elevated, but the people there, I think, are open to that kind of collaboration. So it's not impossible.
Okay, well, thanks for that sort of like, high level landscape view.
Yeah, no worries. Happy to chat more about it. Is it boring GWAS, or is there more things to find? So I think there's this broader idea in, at least in the type 2 diabetes perspective, about that diabetes is not one trait, it's a complex trait where there's lots of different types of diabetes, ways you could get to diabetes through different means, like defects in insulin secretion, overt insulin resistance, high levels of MET syndrome, lipodystrophy. And so one of the things we've tried to do is to actually try to characterize that subtype by genetic physiology. So what we can do is take all of our associations, project a range of cardiometabolic, blood pressure, anthropometric traits onto those associations and try to cluster based on those anthropometric traits and see, are there different axes of diabetes that seem to matter differently for those traits? So on this left graph, you can kind of see that the color of the plot sort of corresponds to the association for type 2 diabetes, and then clustering by the z-scores for the trait that's listed here. So in that first row, it's fasting glucose. So this cluster that involves, that we've labeled here, beta cell plus proinsulin, has high associations with fasting glucose, 2-hour glucose, glycosylated hemoglobin, and proinsulin levels. And that sort of like typifies that cluster. So there's a set of signals associated with diabetes that associate with those traits. Similarly, if you looked at the obesity cluster, there's another set of T2D loci that seem to relate to, waist hip ratio, body mass index, fat percentage, kind of like an anthropometric obesity cluster that doesn't obviously have super high levels of glucose association, and differentiate it from the subtype of those SNPs that matter for that trait. So there's like a couple of clusters that seem to sort of have this distinctive genetic physiological association, which is kind of interesting. And if you project that into sort of like the genetic architecture of what regions of accessible chromatin by different cell type map onto those clusters, what you'd see is that the clusters, for example, that seem to relate to beta cell biology seem to have regions of the genome that are open and accessible in like alpha cells, beta cells, delta cells, basically cell types of the pancreatic islets, versus other clusters have different regions of accessible chromatin in other tissue types. So it seems at this very coarse level that there's physiological cluster relationships to the genetic architecture and what tissues are involved in regulation of genes at those signals. And there's this substructure to the data that seems to map to diabetes risk. So you might think, oh, that's interesting. Can you do something with it? So one hypothesis we tried to test is actually, can those subtypes actually predict different forms of complications for diabetes? So what we can do is take our large-scale GWAS, our genetic risk scores that are partitioned by the clusters that we've identified, and then actually take advantage of a completely independent cohort of individuals from the All of Us study and the Biobank Japan study that didn't contribute to the primary GWAS, and specifically in those cohorts measure different complications of diabetes and ask, can my sort of subcluster of genetic risk for that beta cell biology or that metabolic syndrome predict heart attack or diabetic kidney disease or retinopathy? So here's that plot, and I'll walk through just for ease. Let's pick the furthest one on the left, coronary artery disease. So what I plotted here is basically the effect size of association between the genetic subtype risk score for each one of those subclusters and increased risk of the trait, in this case, coronary artery disease, by that cluster. So what you'll see is that there's in that case, two subclusters, obesity and lipodystrophy, that seem to associate specifically with coronary artery disease risk. So that's kind of nice and interesting because it suggests that perhaps not all pathways that seem to predispose to diabetes risk uniformly predisposed to coronary artery disease risk as a complication, but that maybe a subtype of them, those risk factors do. So if you then walk through the subset of other complications, you can see, well, obesity seems to be a major one across many of them, but perhaps for retinopathy, maybe there's other subtypes of clusters that do matter for that trait. So that, the effect sizes here are relatively modest, but I think what you could imagine at some forms, longer term thinking is, well, if we refine the clusters more, sort of like pinpoint causal variants and expand the nature of the structures of the contributing variants here, maybe we could imagine try to figure out like that fundamental question, like who once they have diabetes, who's at most risk for retinopathy, nephropathy, or heart attack. If we can guess at who those individuals are by these genetic risk scores, then we might be able to like target our interventions on those subjects relative to others, and that might have a translational benefit.
Ben, someone's asking, could you please describe the term on x-axis beta cell plus PI?
Yeah, so those are our sort of like somewhat our attempt to label the clusters based on the physiology that we've described. So for example, beta cell plus PI, the PI stands for proinsulin. So if you go back to the cluster model here, if you look at that first cluster, what you'll see is that between beta cell plus proinsulin and beta cell minus proinsulin is the fact that the first column, type 2 diabetes risk variants are associated with higher proinsulin levels, versus the second cluster, type 2 diabetes risk alleles are associated with lower proinsulin levels. So it seems like it's the distinction between those two is it's similar, except that the fact that one involves different levels of basically proinsulin secretion, circulating levels of proinsulin. So hopefully that makes a little bit of sense.
Yeah, that's good.
Once we followed from that, I think it's also natural to try to, again, in looking to try to tell the story of what individual loci mean. We'd like to know basically, ultimately, like what the target of the gene of the particular association is, and what sort of like pathways molecular physiologically are implicated. And so in some ways, what we'd like to do is say, okay, here's a diabetes signal we think is important, and we'd like to know which ones of them overlap other types of associations for, let's say, molecular traits like expression of genes nearby, or protein level expression, or even expression of circulating levels of metabolites, to try to understand the underlying biology and causes, as well as we might want to like further connect individual signals with specific trait relevant biology like obesity, lipid levels, or insulin levels. So to try to get you a little bit of that intuition here, I have this sort of like picture that I took from this very nice paper from Joe Pickrell a couple years ago that sort of describes this idea of overlap. So on here, I've plotted sort of, is plotted on the x-axis, sort of like genome position. Every one of these dots, let's say, is a SNP association test. And then on the y-axis is sort of the significance of that association. So what we'd like to see is the example on the right, where let's say you've measured the expression of a gene nearby, and you've got this association between one genetic variant and that gene, and it turns out that that same SNP also turns out to be the most associated variant with type 2 diabetes. That suggests there's like nice overlap between the association and this molecular phenotype, maybe even giving you a clue that perhaps the mechanism by which type 2 diabetes changes is through change in gene expression of that nearby transcript. And that's different from, let's say, the example on the left, where actually you do see perhaps a trait or a molecular phenotypic association or a QTL association, but it's in a completely different position from that type 2 diabetes association. So there's really no overlap, even though there might be interesting things going on at the site. So we'd really like to find more examples of the things on the right and throughout the things on the left to try to, again, get an understanding about what the genes are, what the mechanisms are, and maybe biomarkers that predict risk based on that. So again, like many big projects, it involves another collection of like-minded and similar people as part of our global collaborative. And so what we did here, this group did here, is we took our results from our primary GWAS, those almost 1,300 index variants, and when we systematically collected a large number of these molecular phenotype association scans or QTL scans based on either expression, proteomics data, or metabolomics data. And what you'll see is we tried to sample here from a range of tissues we thought were relevant for diabetes risks. So bulk pancreatic islets, adipose tissue, skeletal muscle, liver, hypothalamic tissue. All of these are postulated to be relevant along different axes of diabetes risk. Another thing we tried to do as well is to collect eQTL data, so data related to expression, gene expression, transcript level expression, from populations that are explicitly non-European, thinking that perhaps, even though it's not perhaps the right tissue, that we might be able to capture effects observed in those groups that we might have missed if the tissues we sampled were actually from Europe, given that our study was actually multi-ancestry. So we'll talk about one or two of those examples ultimately. So we did this sort of like overlap analysis. And for the mavens in the audience that know this, there's a tool that helps you do this called Colloq. So we ran this tool across all of these QTL resources, traits, and identified over 12,000 places where our index signal overlapped in that sort of way on that example on the right with either a QTL or a trait with pretty reasonable confidence. So to summarize that broadly, here's sort of like an upset plot that kind of describes this. So what I've listed here are sort of like 4 broad swaths of categories, either trait associations with like obesity or other physiological measures that we described earlier on the clustering of the traits, proteomics data or pQTL data, expression of transcript level data or eQTLs, or changes in association between circulating metabolites or metabolite QTLs. And then on each one of the sort of columns represents sort of like the number of instances where there's a localization signal in the places where there's the colored dot. So in that sort of like second column is probably an interesting one where we have a co-localization with 73 signals that involve a physiological trait, a protein level expression, a transcript level expression, and a metabolite. And you can see that different signals have different overlaps with one or more of these sort of markers. And just to kind of summarize, ultimately, we've got over 2,000 transcripts or genes implicated by either proteomics data or expression data, 730 or so metabolites implicated, and over 60% of our index variants have some kind of co-localization. So we're able to get a little bit more about molecular clues about what these signals are doing. To give you one example to try to whet your appetite a little bit about this, here's one example, and it involves a gene locus called the secretin receptor. So on the right hand, on the left hand side, here's the signal of association in our type 2 diabetes effect, our scan. So pretty healthily associated. Here in this plot on the x-axis is just physical position, on the y-axis again distance of association. So you can see there's like some signal in action here. On the right hand side, I've got sort of like on the top a summary of that data from that LocaZoom plot on the left, and on the bottom I have expression from the Tiger consortium. So that's basically our bulk pancreatic islet association eQTL data. And so what you can see is that a nice example of that sort of cartoon version on the right, where we've got a case where the association signal for the QTL effect implicating secretin receptor maps basically completely and perfectly onto the type 2 diabetes association. So it kind of leads to this hypothesis, oh, well, could it be that change in expression of secretin receptor is associated with change of risk of diabetes? And if you try to paint that out, it turns out like the variant that increases expression of secretin receptor is risk increasing for type 2 diabetes. So like, of course, many genomics experiments, like I haven't studied secretin receptor for my entire life. And so like many blind genomicists stumbling into genomic discovery, we're like, well, what the heck does this do? So here's a couple of details about it in general. So if you don't know anything about secretin, basically secretin receptor is the receptor that binds secretin. Secretin's major physiological action is basically to stimulate pancreatic fluid and bicarbonate secretion. Secretin itself is emitted in the small intestine. It's stimulated to release by gastric acid. And basically what it does in the pancreas is it increases pancreatic and biliary bicarbonate, and ultimately decreases protein acid levels. So it's basically like modulating pH locally. And the thought is that that's supposed to be important for a number of processes, including fat digestion, and just creating a more neutral environment. Secretin itself has a lot of roles in fluid homeostasis, appetite control, and lipolysis. So it's theoretically like in the right ballpark of things that could be related to diabetes risk, but I don't think we know fully the relationship between the hormone itself, the receptor, which is highly expressed in both pancreatic islets and whole pancreas, as well as kidney and lung, and ultimately the relationship to disease. So I think this is like one of those examples where it's like, okay, human genetics focuses a lens on a region of genome that connects to a specific gene and specific biology. So now it's sort of like, okay, maybe this is a place we could study further functionally experimentally to understand exactly what's going on. Let me give you another example, again, sort of focused on sort of the importance of studying diversity in sort of like our QTL resources. So again, in this case, we pull together tissues that, are obviously aren't directly relevant to type 2, but they're pretty large in their sample sizes relative to the tissue-specific ones. So the idea, I think, here is a statistical question one. We'd like to discover QTL effects, so genetic variants that change expression, proteomics expression in a relevant tissue, we think, for diabetes, but we might be able to discover those effects in other tissues just because we have more samples and it's easy to access those tissues. And so here there's collections for Mexican Americans, Puerto Ricans, African Americans. So the goal here is like, what if there are variants that are actually at high frequencies in this population, but at lower frequencies in Europe? Could we discover QTL effects in those populations that we might have missed because we didn't study them? So here's that distribution. You can see that there's a lot of discovery here that actually involve unique mappings to these discovered panels. So basically the answer is there are effects that we can find in these populations with these sort of off-tissue effects that we don't discover if we look at the ones that are tissue specific. And that they're specific to these population groups. So that's great. So there's like this increased reservoir of discoveries that can be potentially made. So to put a point on that, what we found was that 20%, about a quarter of our co-localizations are found in these data sets, but aren't found in the European QTL resources. So emphasizing the idea that we're finding things in these population sets. They're not monophyletic. Just to give you an example of like how that could be, here's one example that maps to chromosome 11. It's nearby a very well-known locus, the CDKN1C locus. On the right-hand side, what you can see is sort of like the expression, or on the top panel here is the locus zoom plot where you can see our association for type 2. It's off the charts. And then if you look at blood QTL data, there's a cisqTL for this gene in the Mexican American cohort that aligns basically perfectly with our type 2 diabetes signal. You might say, okay, well, this is a massive signal. Why did you find it here and not others? So I've highlighted here the allele frequency of where our discovery comes from in our cohort. You can see that basically this variant is really quite common in East Asians and the Americans and has pretty significant associations here. But actually in Europe, it's much less frequent. It's about 5% versus 22 or 37%. So even in our European cohort, we can see a discovered effect. And it's just because we have like millions of samples from Europe for that discovery. But if you look at the eQTL data, that variant is still at 5% frequency. So then if you're trying to ask, well, is it an eQTL in Europeans? You would look at the associations here and say, oh, actually, it doesn't look like it's associated. In contrast, the other cohorts where you do see the variant at high frequency, like in Mexican Americans, you do see this expression level effect. So makes sense. It's sort of statistically intuitive. We have power to discover the eQTL effect because it's frequent in those populations, and we miss it when we don't have the power outside of them. So sort of gives you the value added of why we should continue to fight the fight of expanding diversities across all of our genetic collections, because we can discover things and map them in this way that is pretty sensible. This gene is of course pretty relevant. It's seems like it is involved in beta cell biology and sort of growth of cells. So the gene itself is a pretty good candidate and makes a lot of sense. So I think biologically is intuitive and is a good candidate gene. So I've spent a lot of time like focused on the pancreas, and I don't have to tell this room that this tissue is of course incredibly complicated. So it's easy to think about it as one tissue, but if you zoom in on islets, of course there are many cell types that are specific, that are intimately involved in hormone secretion, signaling, and regulation of glucose levels and insulin secretion as a response. It involves alpha cells, beta cells, delta cells, tissues of the exocrine and endocrine. Again, I focus thinking about metabolic traits, but if I was a type 1 diabetic, I would say, of course, there's immune infiltration of the pancreas involved in a range of different cell types that are integral to that. And so when you view it from this lens, it makes a lot of sense to say, can we increase the resolution of how we study expression in these sets? Can we identify perhaps the cellular compartments that are specific to alpha cells or beta cells that may relate to diabetes risk in general? And so, because conventionally what we've been doing, so like, for example, that case where we've talked about that I mentioned earlier about the secretin receptor, that was all based on bulk islet data. So basically taking islets, extracting them, and sort of like combining all the cell types together to profile expression. So it like pulls together alpha cells, beta cells, delta cells together. So in our case, we were lucked out and actually we found things. But in general, we might have thought, oh, that we're missing heterogeneity within those individual cell types because cell types probably have their own profile of expression. And so it could be the case in contrast to this bottom row, where actually if we could capture data at the level of single cells, we could collect profiles of alpha cells, of beta cells, of gamma or delta cells, and ask specific questions about does genetic variation associate with change in expression in one or more of those cell types? So here it might be in the bulk tissue, we see an effect, but actually at the level of single cell resolution, it turns out it's really quite specific to beta cells or alpha cells or maybe immune cells. And so armed by that kind of intuition, we have another project ongoing that tries to characterize those single cell profiles. And we're fortunate to take advantage of newly generated data from the Human Pancreas Analysis Project. So if you don't know about that project, it's this really great resource that's being currently generated. Right now, there are the data are sort of like received from pancreatic donors from across the country. I think we've got collections from about 151 donors, of which those donors get subject to a range. The pancreases are collected and a range of omics data generation and molecular biological function of islets are targeted for those samples. So one of those assays turns out to be single cell RNA sequencing. So 106 of those are actually been sequenced and whole genome single cell RNA sequenced and whole genome sequenced. So that allows us the capability to perhaps do our experiments where we're trying to correlate genetic variation with cell type specific expression. The donor populations are actually quite interesting. Of course, they're representative of gender. They span the age range. They're not necessarily specific to type 2. We collect type 1 donors, type 2 donors, and controls. So you'll get age ranges from young to old. And nicely and encouragingly, the representative samples we're collecting are diverse as well. So we can start to think about building, again, diverse representations of our sort of maps of catalogs of genetic variation. As I said, that there are actually all sorts of diabetics here, type 1 and type 2, as well as control. So I think there's nice opportunities to, once these catalogs are generated, to actually relate these sets to all types of diseases related to the pancreas. So, one main question, I think, with any single cell data, it's sort of like the main question you spend a lot of your time thinking about is, can you actually, from the whole set of data across all samples, identify relevant cell types that you think map to the cell populations you're interested in studying? And so on the left-hand side is sort of like our current version of that clustering experiment, where we've taken the profiles of expression for all of these dots, which each dot represents an individual cell, across all the samples. So we've pooled all the samples together here, and then profiled and clustered them based on what we think represents different cell types that we think we've captured from these repositories. So we've here labeled them beta cells, alpha cells, delta cells, gamma cells. And part of the way that we've done that, the clustering here is not supervised. So the clusters generated by the algorithms that do the clustering don't label them. They call it cluster 0, 1, or 2. But when we can go back and actually use marker trait genes to then characterize what we think the clusters are. So for example, this orange or red pink cluster that we think represents beta cells, we think are beta cells because they have high expression of marker genes that we think typify beta cells. Similarly, for alpha cells, those are glucagon secreting cells, and so they have high expression of glucagon. Similarly, for delta cells, high expression of somatostatin, and et cetera, et cetera. So the clusters we think we're labeling and identifying seem to have the right profile of high expression of marker genes that we think are typical, as well as sort of like endocrine and exocrine cell types that we've plotted here. So what we can then do is once we have the cell populations, we can aggregate those cells into pseudobulk expression per individual so that every individual we have in the sample now has a profile of their expression for beta cells, alpha cells, delta cells, et cetera. And then we've also whole genome sequenced them. So now we can ask the question, does genetic variation associate with change in expression in one or more of those cells? Just one other sort of like sanity check for the data, just to give you a sense of what the data looks like. Here's just a representation for alpha, beta, delta, and gamma cells, their proportions. Each column here is an individual, and I've just grouped them, labeled them by type 1, type 2, or non-diabetics. So what you can see here, at least in the type 1 diabetics, not unexpectedly or surprisingly, you can kind of see that the proportion of beta cell populations that are present in these for many of the samples are much smaller. And I suspect those are the older populations of type 1s who basically have low or maybe no beta cells in their system. Some of the ones you can see that actually there's like quite a few beta cells in those groups. Those are either probably like very, very young diabetics who haven't fully lost all their beta cells, but are early in life. And so are facing their challenges there. So this looks representative, nice, gives us an idea that we are representing and measuring cell types in all of our samples, which is nice too. So if we do that experiment and try to identify genes that have cell type specific effects, genetic variants that relate to those genes, here is just, again, an upset plot to describe those genes and what cells they're found in. And so what you can see is that we find many cases of genes where genetic variation associates with change in expression of those genes that are very quite specific to either beta cells, alpha cells, or in this case, delta cells. Those were the three populations we started with that we had the most data in which to do this experiment, ones that we thought we were the best statistically powered to do discovery in. But what you can also see here is that there are many examples of cases where there's actually overlap in those variants, the genes that they alter expression in, and that they implicate more than one cell type. So alpha and beta cells, beta and delta cells, et cetera. So, variants don't have to be cell type specific. They can involve change in gene expression in multiple cell types as well. So, but not unexpectedly, most of those observations are cell type specific. And the difference between, let's say, alpha and beta cells and delta cells here is a difference in power. I think we have twice as many samples used for the analysis of delta cells than we do the other populations, just as a function of our discovery efforts here. So I don't think that's about biology. I think that's just about statistical power for discovery in those cell types. To give you, to try to cut to the chase, can we connect genes that we think are involved in differences in expression in those particular cell types with association signals for diabetes? Here's like a short list of our very preliminary work to do that. And so basically to say, yes, there actually are examples of genes that meet this criteria. Many of these genes, I don't think we would have necessarily prioritized or jumped out at us. Some of these genes I recognize, but like ADCY5 or lamin-A, but many of these are not immediately things that jump out to me. So that actually maybe suggests that there's some new biology we have to chase down a bit. I'll pick one example here that looks relatively promising. NCK1 actually has a little bit of biology on it. So it looks like, here's the violin plot to just show you sort of like the relationship between genetic variation and change in expression. So you can see that the type 2 diabetes allele, one of those alleles definitely changes expression in, I think it's alpha cells in this case. And it looks like there's some model systems work that is at least involved, has focused on liver related traits, so not in pancreas specifically, but it seems to suggest that like knockdown of NK1 seems to improve glucose tolerance and insulin signaling. So there's some biology that suggests that in model systems that this might be relevant to diabetes. But again, I think this is like a motivated case. I don't think this gene has been explored deeply and certainly not explored in the context of the pancreas, even though it's known to be expressed in the pancreas. And in this case, perhaps alpha cell biology might be the place where human genetic variation is implicated. So hopefully what I've relayed to you is sort of like a broad swath, this idea that like diverse discovery is important. We can discover new loci for diabetes. that discovery leads us to understand subtypes of diabetes that help predict complications, that we can use these data to connect to genes that are ultimately responsible, figuring out what ultimately the candidate genes are, and actually that what we should do is collect a diverse ancestry of data for all of these molecular phenotypes as well, because we'll actually make additional discoveries if we do that. And that actually, we can further add to this resource by looking at single cell resources for pancreas and maybe other types of tissues. And actually, if we do and build those catalogs, we can find additional candidate genes for diabetes, all of which kind of centralizes, I think, around a theme about just generally broadening our diversity of who we're studying to make those observations relevant to everyone. So hopefully it gives you kind of like a sense of the roadmap here and a little bit of how I think about it. We're involved in one of the reasons I think I'm thinking about it in this way is that the functional experiments are really hard, and so I'm fortunate to be involved in other consortium where people who study particular cell models, mouse models, zebrafish models, cell culture models are just waiting to try to activate some of the observations to really test molecular physiology very concretely. And so hopefully this kind of effort will contribute to those activities. But that's what I have. Thanks for having me. It was basically everyone else that contributed to this work, not me. You saw their pictures, particularly my graduate student and one of my bioinformatics specialists here helped with this work, but happy to answer other questions as you have them. But thanks for listening.
Thanks, Ben. That was really interesting. And let's open it up for questions. You can drop something in the chat, or you can ask to raise your hand or just unmute yourself. I had a question, just the second to last slide that you had there, that whole pipeline. How can you, just sort of, speculating, how could you, or how do you envision getting all these, these new GWAS discoveries out to those who can actually validate the mechanisms through functional experimentation in model systems? Like, how can you improve that pipeline?
Yeah, I think that, to me, actually, I think that's the hardest part of doing actually all of these experiments. It's something in the group, the consortium that I'm involved in locally. It's just really challenging. So like, I think the best way that I can imagine it is just developing partnerships with people who study particular systems that are accessible and tractable. I think it's one investigator cannot tackle the entire heterogeneity, tissue heterogeneity of diabetes. So if one can build partnerships with people who are really have got medium throughput models for hepatic-like cells that are differentiated from iPSCs, or have CRISPR screening techniques that have focused on cell types that you think are relevant for the aspects of disease that you want to study, I think that's the place where you have to build partnerships and just funnel it through it that way, because, and even those conversations are hard because the molecular biologists, in my experience, are not, they look to the human geneticists for help to try to interpret what things are interesting and what's not interesting. But the human geneticist, like, I wish I was a deep caring molecular biologist that knew everything about like a particular cell model system, because then I could just do that in my lab. But I need the wisdom and expertise of the molecular biologist to understand what is actually tractable as an experiment to chase down. So I think it's really about building those partnerships and finding friends and colleagues and smart people at individual tissues or models, and then trying to figure out and mine out of the data what are the most likely things to matter. And when we've done those kinds of experiments, like on skeletal muscle, finding an expert, and they look at the list of genes and they're like, this looks really interesting. And they have like a list of 5 things or 6 things. And that can really lead to advancement. So that's my current best thinking. But there, it's funding driven, it's interest driven, it's like your community driven
Right.
accessibility,
Yeah, well, you're preaching to the choir here, because collaboration and connection across, interdisciplinary science for this kind of complex disease that is type 1, I think is kind of like our sweet spot, or we're trying to make it our sweet spot. We're trying to, facilitate these kinds of connections because it's just it's such a complex disease and everyone looks at it from a different perspective. But now this whole genetic complement brings so much power. So that's great. I appreciate what you said. Here's a question from Sharma Banerjee. 90% of the GWAS signals in the non-coding regions. How can one focus on the causal variant, which might act as cis-regulatory?
Yeah, this is this is the fundamental challenge. And so I think depending on how you dissect it, there's lots of ways to think about it. I guess for my taste, I think I end up focusing on two things. One, and I didn't get a chance to talk about it, is sort of like the pinpointing of the actual causal variant. So out of all the things that are linked, it's non-coding, but can you narrow down on the subset of variants that you think are really likely to be causal? And that's probably not all of them. It's probably a subset of them. And then, yeah, and then you think, okay, it's non-coding. If it's not a promoter, it's probably a distal enhancer. And so then you start to try to layer in epigenetic resources, chromatin accessibility resources, and it basically like annotations in the genome that tell you and give you a clue about what the tissue of relevance is and what the mechanism is. And then again, try to use, molecular phenotype data like expression QTL or protein QTL to try to link that non-coding variant to a candidate gene. Because once you have a candidate gene, you can activate like a model system. You can do mouse experiments, you could do fly or zebrafish experiments, you could do cell culture experiments, knocking the gene out. That's how I think about it. I know that there's a lot of vibrancy around doing CRISPR screening on enhancers directly. So you really want to try to understand whether or not the enhancer matters and what gene it knocks out. And those experiments are nice, like CRISPR-I experiments, to like basically situate a CRISPR on the putative enhancer and see if it changes expression of a gene nearby, and if so, what gene it is. And the advantage of those experiments is you can do them relatively at high throughput, because you're probably not interested in testing one enhancer element. You've probably got like dozens or maybe hundreds you want to test. And so if you've got the right tissue and model, you might make some progress about figuring out what the activity is and then what gene it connects to. So I know lots of people think about that. And that's, I think, a really fruitful line as well.
Great answer. Ruby Sharma.
A very nice talk. I just wanted to know one thing. Like you told about the alpha cells are equal, like more than the beta cells in both the type 2 diabetes and the type 1 diabetes. But yesterday only I was listening to Alvin Paz and he was telling like when there's a destruction of the beta cells, there is like being a neighbor, it secretes some of the cytokines and there is a reduction in the alpha cells also, and thus the glucagon level also gets changed. So would you like to say something about it?
No, I think that's really interesting. And I think that's the kind of the promise of trying to study trying to collect data at single cell resolution in diverse subjects with diverse phenotypes. I think gives us a chance to really tease out exactly those kinds of changes. Maybe even if you collect individuals over time, you can even see what those changes are fundamentally. Like maybe things are more complicated than just like beta cell destruction in type 1. Maybe there's some other competing forces that we didn't appreciate, but by collecting data and sort of like trying to do correlation and physiological work, we can actually try to think about that more carefully. So I'm cautiously optimistic we'll be able to think about that just by the data collection and doing some analysis there.
Thank you.
Yeah, there's so much room for inquiry and exploration in this realm. I wondered, any more questions? Anyone else like to ask a question? Okay, last question for you, Ben. Looking for postdocs, looking for anyone to join your laboratory at this point? Looks like
always and constantly. Yeah, I mean, we're
a lot of work to be done.
Yeah, there is, and we think a lot about it, lots of different aspects, assembling data, doing human genetics, trying to build machine learning models, maybe even doing data collection. So there's lots of things to be done, lots of opportunities to be had.
Right. So if anyone has interest in this realm, please reach out to Ben directly. And Ben, thank you so much for walking us through your work at a very high level and, kind of bringing it to bear against the T1D questions, that a lot of the audience are pursuing currently. I hope you
Yeah.
have a great rest of the day, and thanks again for joining.
Yeah, thanks for having me. Appreciate it. Cheers.
Okay, bye-bye.